Many thanks for joining us for our first Milner seminar of the year. It's a great pleasure today to have Professor Ziad Mallat speaking. Ziad is the BHF Professor of Cardiovascular Medicine here at the University of Cambridge, where his group has been based, I think, since 2010. And I understand that he will also be moving this year into the new Heart and Lung Research Institute. And previous to this, his research group was based at INSERM in Paris, where he also trained as a clinician scientist in the 90s. And his long-term focus on really understanding atherosclerosis as a disease of chronic inflammation has really started to help unraveling some of the immunology underlying this disease, its development and potential strategies for prevention. And his contributions to this field were recognized in 2020 when he was elected as a Fellow of the Academy of Medical Sciences. So it's a great pleasure to have you, Ziad, and I'll hand over now to you for the talk. Many thanks.
Yeah, thank you very much for inviting me to give this talk. I will start by reviewing very quickly the basis, what is atherosclerosis and ischemic heart disease very quickly, and how inflammation has been recognized to play a role in this, and give you the rationale for the early phase clinical translational studies that you are doing in Cambridge, trying to put in place immunomodulatory therapies for to benefit potentially patients with ischemic heart disease. So, atherosclerosis is really the major cause of disability and premature death in the world. And atherosclerosis, in fact, is due to blockage of the artery by the buildup of initially fatty material that grow up to limit blood flow and oxygen supply to the major organs. These plaques can even break off and lead to clot formation, and this may therefore cause complete blockage. And if this happens, in the arteries that supply the heart, you have a heart attack in the brain, it's stroke, and it can be happening everywhere in the body. And heart attacks and strokes are major responsible for heart failure and vascular dementia, and are very prevalent still, despite really the best of our current treatment. And we think this is for a reason, and there should be a missing link. And we really think that this is still happening because we do not treat the root cause of atherosclerosis, which is a chronic inflammation of the arteries and a malfunctioning of the immune system. In fact, when these initial fatty deposits start to accumulate in the artery wall, they create a response of the immune system, which sends a lot of immune cells there to try to clear the abnormal material. And this is a more scientific picture of what we believe is happening in the artery wall. So when these LDL cholesterol start to accumulate from the blood, start to accumulate in the artery wall here, the intima, it becomes modified and oxidized. And this oxidation make these lipoprotein particles recognizable by the immune system. They are recognizable by immune receptors. Here we put scavenger receptors like SRA or CD36, but also innate immune receptors on the vascular cells themselves, on the lining of the arteries, endothelial cells, leading to expression of chemokines and adhesion molecules and the recruitment of inflammatory cells like here, monocytes, coming into the artery initially to differentiate into macrophages and take up the lipids. So initially, it is suggested that this is a beneficial response in order to clear the artery wall. However, quite often really, this goes on to create more inflammation. So the activation of innate immune receptors, as shown here, create more production of inflammatory mediators that activate more the artery wall to bring more inflammatory cells. And now we know a lot due to, and thanks to really single cell studies on mouse atherosclerotic lesions and human atherosclerotic lesions, we know a lot about the different cell subtypes and immune cells that accumulate in the artery wall, different subtypes of monocytes and macrophages, but also different subtypes of lymphocytes, T cells, B cells, etc. And this is one example, for example, of the variety of T cells that you can find in human atherosclerotic lesions of people with stroke, for example. This is from carotid endarterectomy plaques for people undergoing surgery of their plaques after stroke. And there is a huge variety of cells that can induce inflammatory response, interacts with each others and inflame the artery that goes on. So this is a description of the variety of innate and adaptive immune cells that we can find in atherosclerosis. But they are not just bystander accumulation of inflammatory cells. In fact, we know from different sources and experiments that inflammation is mandatory step for developing to develop atherosclerosis. This is an obligatory step. This is from an experimental work in mice, where you have mice under high cholesterol and with very high cholesterol levels in the blood. If these mice do not have monocytes, despite the high levels of cholesterol, they do not form any atherosclerosis. And this is true for T cells and B cells for the absence of T cells and B cells. So it's not just you need risk factors in order to induce the disease. These factors, in fact, induce the disease through the induction of a chronic inflammation. And if you know how this chronic inflammatory response is being regulated, then you can potentially substantially impact on disease development. So this is in mice and experimentally speaking, but also we have data now from genome-wide association studies of coronary artery disease that identified inflammation-related genetic variants that are thought to associated causally to the disease. And I put here a few examples like the IL-6 receptor signaling pathway, interleukin-5. This is another chemokines here. These are adhesion molecules that are expected to be causally related to the disease. So there's now an important evidence that inflammation played a role in atherosclerotic disease. And despite this, we still have no anti-inflammatory therapy to treat patients with ischemic heart disease. And this is not because nobody has tried it. I mean, there are studies that have been conducted to test anti-inflammatory therapies in cardiovascular disease. And I will give you just few examples here with canakinumab, which is a monoclonal antibody against interleukin-1 beta, underwent a phase 3 clinical trial in patients with post myocardial infarction and with stable coronary artery disease. It has shown benefit on reduction of cardiovascular events by 10 to 15%. However, it has not been approved for use because of the generation of quite important side effects like increase in the risk of fatal infection. So we need to know how to modulate and manipulate the immune system in order to get benefit from its modulation, but not induce any potential side effects. Now, other therapies are being still trialed, like anti-IL-1 blockade of IL-1 alpha, blockade of the IL-6 pathway, and other therapies are in development, but not yet in clinical trials. What I would like to present to you here is two different modalities of modulating the immune response, targeting Treg cells, regulatory T cells, and B cells. And I will start first by showing you what is the rationale for this and how we are now moving with these therapies into clinical trials in patients with ischemic heart disease in Cambridge, and with the aim potentially to test them and to see whether they can benefit patients with ischemic heart disease. So the first thing is the rationale for use of interleukin-2 initially is based on an important role of adaptive immune system in promoting the development and progression of atherosclerosis. We know from several studies that adaptive T cells are involved in the progression of atherosclerosis. They are They respond to antigens loaded on major histocompatibility complexes and respond in a specific manner and can be activated to give help to B cells to produce antibodies against these antigens. And most of, what prototypical antigens in atherosclerosis that can be processed by antigen-presenting cells like dendritic cells are antigens that can come from lipoproteins, either native lipoproteins or oxidized modified lipoproteins. And in fact, it's just a summary of view of what we know about this, is that when these oxidized lipoproteins are taken up by antigen-presenting cells, they are activating immune receptors on these antigen-presenting cells. And this presentation of antigen is so happening in a pro-inflammatory environment. So this is leading to the induction of co-stimulatory molecules on these antigen-presenting cells and leading to the differentiation of T cells towards helper T cells, for example, for CD4, essentially in atherosclerosis, in the setting of atherosclerosis, Th1 producing interferon gamma and Th17 producing IL-17 molecules, but also activating CD8 T cells to become cytotoxic T cells and producing also interferon gamma. And all these pathways have been shown in experimental models of atherosclerosis to be pro-atherogenic. But now what we know, in fact, is that initially at the start of the process, you start from a healthy situation. And what we know is that in a healthy situation, native lipoprotein particles that are taken up by antigen-presenting cells, in fact, are presenting antigens in a relatively non-inflammatory milieu. And this is leading to the generation of regulatory T cells that could be specific for these peptides derived from these lipoproteins. And we know that these Tregs, with their potential immunosuppressive and regulatory properties like production of IL-10 and TGF-beta can prevent against the development and progression of atherosclerosis. And this has been shown by us and many others in various models of the disease. And what we think now is that initially we develop a regulatory immune response. And when this regulatory immune response is broken down, and then this would lead then to activation of adaptive effector T cell responses. And in fact, there is data here, for example, an example from Klaus Ley group of data showing detecting in the circulating blood of human beings CD4 T cells that recognize MHC class II restricted epitopes from apolipoprotein B, which is the major component of LDL. They are able to detect this using specific tetramer. And as you can see here, in the absence of cardiovascular disease, in individuals with no clinical manifestation of cardiovascular disease, most of these CD4 T cells that recognize these epitopes are express FOXP3, and probably they are regulatory. And then they switch towards a Th1 and Th17 phenotype with disease progression. So what we think is, and there are other studies, showing supporting these things, is that there is quite a deficiency in the regulatory arm of the immune response in patients with with ischemic heart disease, like here, non-ST elevation myocardial infarction patients, in comparison with patients with stable coronary artery disease or control patients. Even the total numbers is reduced in addition to their functions. And we know that these circulating numbers of Tregs, in the blood of individuals are inversely related to the risk of developing myocardial infarction in the future. And this is from an important studies from Sweden with many individuals included there. So these studies in mice and humans really suggest that modulating the regulatory arm of the immune response will be essential in limiting inflammation and controlling inflammation during atherosclerosis and potentially limiting cardiovascular events. So one really interesting way we thought that it would be nice and interesting to try is is using the properties of interleukin-2 to modulate selectively T effector cells on one side and Tregs on the other side, dependent on the dose of interleukin-2. This is shown here in a very simplistic manner, let's say. You can see here in order to activate effector T cells here in red, you would need concentrations of interleukin-2 that are around here in vitro, around thousands of international unit per mil, in order to activate phosphorylation of this STAT5 transcription factors that is downstream of IL-2 signaling pathway. However, as you can see here, thousandfold lower doses of interleukin-2 are able to activate the Tregs without activating any effector T cells. So this property of the exquisite sensitivity of Tregs to low doses of interleukin-2 is now being harnessed for the treatment of autoimmune diseases. And the idea here is that in chronic inflammation and autoimmunity, there is an imbalance of the immune response towards less Tregs and more T effectors, and low doses of interleukin-2 can be interesting to use in order to reestablish a balance in the immune response, increase Tregs and their suppressive properties, and try to resolve the chronic inflammation there. So we wanted to test this in patients with cardiovascular disease. However, the currently approved interleukin-2 is approved at high doses for the treatment of metastatic renal cancer, and high doses of interleukin-2 are known to potentially may induce some important side effects like capillary leak syndrome, and the use of interleukin-2 is contraindicated in the setting of ischemic heart disease. So we needed to start by a safety study in patients with stable coronary artery disease using low dose interleukin-2 in order to show safety, and then move into a biological efficacy trial to see whether low dose interleukin-2 in these individuals, not only whether it's safe, but whether it can increase Tregs in a selective manner without inducing side effects, inducing T effector cells or other kind of activation of the immune system. So we designed a study with two parts, part A and part B, and first part A with stable ischemic heart disease patients, and tested escalating doses of interleukin-2 within the range of interleukin-2 that was shown in other chronic inflammatory disease, that it might be selective for induction of Tregs over T effectors. And once we have finalized this part A, we moved on to a part B in patients with acute coronary syndromes, and testing based on the data from part A, 2 doses to see whether, again, they are safe and whether they are selective for Tregs. So this study has been recently published in NEJM Evidence, and you can go and look at the details of the data. This is a summary of what you have obtained, is that there is we have seen no serious adverse events that are related to the drug. And most of the common adverse events that we see are transient and mild adverse event related to the injection of the recombinant low-dose interleukin-2. In terms of biological efficacy on Tregs and T effectors, this is the part A of the study in patients with stable coronary artery disease. And you can see here the different doses, the range of IL-2 that has been given to these patients. In fact, what we have done, and sorry, I've not mentioned that, is that we are giving them daily doses, daily injections for 5 consecutive days, and addressing an induction dose, an induction phase, and addressing their Tregs and T effectors and other, studying their immune system, baseline and at day 6, so after the induction dose, but also 1 week after stopping the injection of IL-2. So you can see here the dose, let's say dose-dependent increase in Tregs in these stable angina patients without any change in their T effectors. And in the acute coronary syndrome patients, these two doses of 1.5 and 2.5 million units per day also led to a very substantial increase in Tregs, around 100% increase, without again touching on the effect on the T effectors. However, we wanted to have more information, and early phase clinical trials should be designed to do really very careful and detailed mechanistic studies in order to de-risk any subsequent clinical phase trials. And so we wanted to really address the impact of these doses of IL-2 on the immune response in a detailed manner. This you can see here from flow cytometry data that at day 6 after the induction dose, both doses of 1.5 and 2.5 selectively induced Tregs. However, as you can see here at day 13, so 1 week after stopping the dose of IL-2, the 2.5 has also led to some increases in T effector cells, T central memory, T effector memory cells, and some NK cell increase. So we wanted to have more detailed information about what's happening, and we collaborated with the group of Mena Clatworthy here in Cambridge, to do single cell RNA sequencing in CITE-seq, in fact, on these individuals with acute coronary syndromes at baseline and after the treatment, the induction dose of IL-2, and comparing patients who received placebo to patients who received the 1.5 and the 2.5 million dose. And this is a summary of what we find. This is a UMAP projection of the different cell types that you can find in the blood of these patients, the different distributions. And we confirmed, of course, that Tregs were induced significantly by both the 1.5 dose and the 2.5 dose in comparison with placebo. But as you can see here also, we confirmed that the 2.5 dose had a tendency, significant way to increase also T central memory cells, reduce the naive T cell population. So in more detailed analysis, we also addressed the relationship between the Treg expansion and the expansion also the Treg clonotype size. And this is the clonotype size on top of the UMAP projection. And you can see here for the 1.5 dose, for example, there is an increase in the clonotype size of the Tregs here, as you can see here with the 1.5 and even more with the 2.5. However, you can also see that the 2.5 dose is also increasing the clonotype size of the T effector memory cells. So this could be potentially, we don't know, but potentially detrimental. The other thing is that when we look at the different pathways that are differentially regulated between the different subtypes of T cells, Tregs, effector memory, and central memory, and compare the two different doses, 1.5 in blue and the 2.5 in orange here, you can see that the 1.5 dose is really very selective for the Tregs and inducing differential activation of differential pathways selectively in Tregs in comparison with the T effector and central memory T cells. So based on all of these data and other data that I'll not show you here, that we decided now to go with the 1.5 million dose into a phase 2b clinical trial that is currently ongoing, which is the Ivory trial, and dosing this and testing the impact of this 1.5 million units on the reduction of vascular inflammation in patients with acute coronary syndromes, and this is currently ongoing. Randomization of these individuals, they have PET, they have baseline FDG PET CT in order to look at their vascular inflammation at baseline, randomized to low dose and to look into a placebo for an 8-week treatment period, and a post-dose PET CT imaging in order to have an idea about how this IL-2 can impact their vascular inflammation. And I hope I can talk to you about the results of this next year. Now, learning from this, I mean, going forward, we have shown, I mean, we have obtained really interesting data. For example, we know that probably IL-2 will also be impactful on other protective immune cell subtypes. This is a study we were doing in mice and myocardial infarction and looking at the impact on the role of innate lymphoid cells type 2. These are cells that produce type 2 cytokines like IL-5 and IL-13 that are important in myocardial repair. We have shown that they are important in myocardial repair. And during our analysis, we obtained unbiased data to suggest that IL-2 could be an upstream activator also of these innate lymphoid cells type 2. And supplementation of IL-2 in these mice after myocardial infarction improves their recovery of heart function. And in fact, we wondered whether this is also true in human beings. So we built on this LILAC trial that we were doing in patients with acute coronary syndromes. And in fact, really nicely, we can see that this low dose of IL-2, whether the 1.5 or the 2.5, can really activate interleukin innate lymphoid cells type 2 in vivo, as shown here, but one marker, the increase in the production of IL-5 in the blood. So what we really expect from this low dose IL-2 in patients with acute coronary syndromes is that it will promote Treg activation, leading to IL-10 and TG beta-mediated resolution of inflammation, but potentially also activating your repair mechanisms through activation of these innate lymphoid cells type 2 and leading to resolution of inflammation. The next, oh, sorry, I don't know what's happening here. Okay. The second part of my talk will be briefly about the other type of modulation of inflammation that we are pursuing in patients with myocardial infarction, and this is about the regulation of the B cell responses. As I told you, these B cells are also involved in atherosclerotic disease and myocardial infarction, and I will show you some data. And in fact, this is not true only in mice. We know also, again, from genome-wide association studies and systems biology approaches of these gene network, that modules involving B cell activation, differentiation, B cell receptor signaling pathways are really enriched in these networks that are potentially causally associated with coronary artery disease in human beings. And we and others have shown using a series of experimental studies that different types of B cells are involved in the regulation of the development of atherosclerosis. I will not go through all these studies, but we know, for example, that depletion of these mature B cells, the follicular type of B cells, is important in limiting the progression of atherosclerosis in mouse models, and this has been confirmed by many other groups. So this pathway of mature B2 cell activation, generation of germinal center B cell responses and antibodies against, for example, as I said, LDL-derived antigens, is detrimental in atherosclerosis. And if you can manipulate this pathway, you could potentially reduce the initiation and progression of atherosclerosis. What is even also interesting is that in the setting of myocardial infarction, the release of necrotic debris has been shown by us and others to activate also B cells to produce chemokines like CCL7 and orchestrate the recruitment of innate monocytes, Ly6E high monocytes, inflammatory monocytes to the inflammatory sites. And this is, we have shown that this is very detrimental. And if we deplete B cells at the acute phase of myocardial infarction, we are able to reduce the detrimental myocardial remodeling post-MI and improve the recovery of heart function. So B cells would be important in atherosclerosis development, but also in the acute response to myocardial ischemic injury. And in fact, a very recent study by an Australian group has shown that that B cells could also be important in the acceleration of atherosclerosis post-myocardial infarction. We know from other studies that when patients get myocardial infarction, this is a potential risk for also further acceleration of their atherosclerotic process. And this group has recently shown that after myocardial infarction, a kind of damage associated molecular patterns activate B cells and leads to the activation of the germinal center B cell response in the spleen, leading to formation of plasma cells and antibodies in the circulation. We don't know exactly now what are the specificities of these autoantibodies, but this has been shown by these groups to be important in the acceleration of atherosclerosis post-MI. So our hypothesis here is that in post-MI setting, depletion of these B cells could be an interesting way in order to limit this deleterious response and improve the remodeling of the heart post-MI, and in the same time limit the progression of atherosclerosis post-MI. And in fact, we are working with drugs that exist already and trying to repurpose them. So we decided to use rituximab, which is an anti-CD20 antibody that is being used by millions of patients, particularly in patients with lymphoma and other chronic inflammatory and autoimmune diseases like RA and lupus. But again, in atherosclerosis and myocardial infarction, it is contraindicated for unknown reasons. So we again needed to go through a phase 1, 2A safety clinical trials in order to test the safety and biological efficacy of treating patients with ST elevation MI with rituximab and see whether it's safe and whether it does its job in reducing acutely and very quickly depleting B cells from the circulation. So these were our inclusion criteria at all patients with ST elevation MI with successful primary dilatation and stenting of their arteries, and excluding any patients with very severe disease at the beginning or with infections. And we tested, in fact, 4 different doses of rituximab. The dose of rituximab that is being used in patients with rheumatoid arthritis and SLE, for example, is around 1 gram per infusion repetitively. However, we wanted to dose the patients with lower doses to see whether we can still have biological efficacy while potentially reducing any potential side effects. So these 4 doses were tested in these patients. And in fact, this has been also published recently. We have not seen any really serious adverse events related to rituximab injection at the acute phase of MI. And just look at this part A of the panels here with the different doses of rituximab infused. So this is one single infusion. You can see that even with a 200 milligram dose at 30 minutes after the start of the infusion, we deplete most of the B cells in the blood, and this is sustained for a few months with the highest doses. But we have seen some recovery of these B cells with the lower doses. And in fact, the B cells that recover in the lower doses are mostly of transitional B cell phenotype, which could have, in fact, regulatory properties. I would be happy to discuss this later on. So again, and again, when we looked at left ventricular ejection fraction in these patients between the index and follow-up, we have seen some trend towards an increase. So again, a good safety signals at least in these individuals. So we have now moved with the phase 2B clinical trial, and this is a European multi-center clinical trial that is funded by the European Union. And the aim of this, and this is starting now, is to test 2 different doses of rituximab. The lowest doses that we tested, 200 milligrams versus the highest dose, 1 gram, against placebo in patients with acute ST elevation myocardial infarction. And the aim of this is to assess the impact of this therapy on the recovery of left ventricular ejection fraction as measured by cardiac magnetic resonance imaging at 6 months follow-up. And of course, there are other secondary aim to this, looking at impact of rituximab on infarct size, but also a nested sub-study in this, looking at the impact of rituximab on vascular inflammation using, as I said, for the IL-2 trial, FDG PET-CT imaging. So this is currently ongoing and including enough numbers of patients in order to be able to detect 5% change in left ventricular ejection fraction, which we think could be enough in order to say that this is relevant clinically to limit potentially the detrimental cardiac remodeling. So in conclusion, I hope I have shown you that lymphocytes, that atherosclerosis is not just a lipid-mediated disease, that the immune system is necessary for its development and complications, and lymphocytes are major contributors. Tregs and innate lymphocyte cells type 2 are potent anti-atherogenic and cardioprotective immune cells, and low dose interleukin-2 is safe and well tolerated in patients with acute coronary syndromes. It selectively activates Tregs, but also, in fact, in the protective innate lymphocyte cell type 2, and we believe that it is really, this is an interesting double hit strategy in order to modulate inflammation there in these patients. B cells, and particularly these follicular B cells and germinal center B cells, are pro-atherogenic and are deleterious for post-MI cardiac remodeling. And their depletion with rituximab appears to be safe. And now we are testing whether this could be beneficial in phase 2b clinical trials in patients with acute ischemic coronary syndromes. Just to finish, in fact, and just to show you is that we are thinking about improving all of this, because I mean these are therapies that already exist and we needed to repurpose them and see what happens. However, we are thinking about more targeted therapies and developing these currently. For example, we have developed an IL-2 fusion molecule with a single chain antibody that recognize components of the atherosclerotic artery wall. And the idea here is that this fusion molecule will be particularly enriched in the artery wall and induce local expansion of Tregs and limiting any potential other side effects that could be envisaged. And this work is done with David Klutzman, Alan Tedge in France, and Christoph Bender in Austria. And other things, we are collaborating with Willem Mulder in the Netherlands and Robin Chattock on strategies to target the delivery of RNAs or siRNAs to specific types of B cells or T cells in vivo and modulate the specific pathways that we see are important in disease progression. And I hope that in the next years, I will be able to show you data on this. This work would never been possible without all my great collaborators. And I would like here to thank particularly Tian Zhao, a clinical lecturer in my lab, in my group, who is leading on these clinical translational studies, with Steve Hull and Papworth as the chief investigators on the B-cell rituximab studies, and Joseph Charon as chief investigators on the IL-2 studies, and with really very important collaboration with Mena Clatworthy here in Cambridge. Thank you very much for your attention.